We are all different. Our hair, skin, eyes and height are just some of the many features which may vary. Each of us is unique. Yet people in the same family often look alike. For instance, brothers and sisters can have the same colouring and features, and children often resemble their parents. It's the same with other animals. Even very young offspring have the same shape and markings as their parents. So it seems that characteristics can be inherited or passed on from parents to children. Genetics is the study of how inheritance happens. At a major plant research institute in Norwich, the genetics of pea plants have been looked at in detail. Peas have a number of characteristics which can be inherited.
Well, I could spend hours talking to you about the differences in peas, but if you start with the most obvious differences, you'll find tall peas and short peas, peas with white flowers, peas with coloured flowers. You'll find peas with green pods, with yellow pods, you name it, we have it.
But how are these different characteristics passed on from generation to generation?
Well, the process is affected by genes, and we have genes for all the characters that we've been talking about, so genes for red flowers and genes for white flowers.
In plant and animal cells are thread-like structures called chromosomes. They carry a set of plans which tell the cell how to develop. The chromosomes are made up of genes, and the genes control individual characteristics. Normally, chromosomes exist in pairs, but both the female and male sex cells contain only one set of chromosomes. So when fertilization happens, the parent cells pass on single chromosomes to form pairs again in the offspring. That's how chromosomes and characteristics are inherited. How could this be shown in peas?
A very good example, and a simple one, would be to study the differences in flower colour in peas. If you decide to cross, say, a red-flowered and a white-flowered pea, this will be a very simple thing to do. And you'd have to go into the field and collect some flowers from the, say, the red-flowered plant. Bring these back into the glasshouse and then prepare the white-flowered plant as the female. A glasshouse is the best environment to do it because you are free from damage by rain, you're free from attacks by birds, and even the possibilities of pollination by insects.
Unlike humans, each flower has both male and female parts. Brian takes a very young white flower and carefully removes all the yellow pollen sacs, the male parts, so it can't fertilize itself. Only the female part, or stigma, remains. From the male pollen sacs of the red flower, he scoops out some pollen to place on the stigma of the white flower. The male cells from the red flower fertilize the female cells of the white flower. A pea pod then develops, and the pea seeds can be planted out to produce the next generation of pea plants. But what does crossing red and white flowers produce?
Crossing a red-flowered pea with a white-flowered pea, the first generation, or F1 as it's known, you'll find, without exception, the pea flowers are red. There are no white-flowered forms at all. They are red, and they look exactly like the red-flowered parent that we used in the first place.
But what about the genes inside those red flowers?
Genetically, it's a different situation. They may look to you and to me and to everybody else as red flowers, which of course they are. But genetically, you have two genes, remember, one from the male cell and one from the female cell. One is a red gene and one is a white gene. And because red is dominant, we only see the red. It is overpowering the white gene, if you like, or it's switched on, whereas the white gene is not. And so this dominance means that the white gene might as well not be there. There is no dilution, that's why you don't get the pink flower. The dominance leads to the full expression of red colour, and no other colour is allowed, if you like, to develop.
The pure red-flowered parent has a pair of identical genes, both instructing the flower to be red. The pure white parent has two identical genes instructing it to be white. Each parent passes one gene on to the next generation, so, however these combine, every new plant must contain one red and one white gene. But all the first generation of plants look red, so the red gene is dominant. The white gene has no effect on the flower colour. It's called a recessive gene. But now what happens when these first-generation plants reproduce?
Now, when you allow that particular first-generation plant to self-pollinate, then these genes will separate. And you'll find that if you look at the next generation, which is called the F2 or second generation, you will not have what you had in the first generation. You will have a mixture of red-flowered peas and white-flowered peas. Always, though, a majority of flowers which are red. You'll find that the relationship between the red and white flowers is very constant, and it approximates to a 3 red to 1 white ratio.
The first generation plants which we see here all carry 1 red and 1 white gene. If a red gene from a male cell pairs with a red gene from a female cell, pure red flowers are produced. If 1 red and 1 white gene come together, these plants will be red, as red is dominant. But if both pass on white genes, a pure white flower will be produced. The odds are that 3 out of 4 will be red flowers and 1 will be white. But why study genetics in peas?
Well, mainly because we're trying to improve the pea as a crop. We're very short of vegetable protein in this country. We rely tremendously upon our imports from the United States of America and imports from around the world generally, which is a bad thing to do. So we feel that within Northern Europe in particular, we should improve a high-protein crop.
Genetics is useful in many areas of farming. Herefordshire is the home of the Hereford breed of cattle. In fact, the first Hereford herd in the world was bred here.
There have been Herefords bred on this farm for over 200 years. And we have been breeding them here for about 60 years.
The Hereford bull and cows all show particular characteristics.
The Hereford is a white face with a rich red body, good heavy legs, big boned legs, and a good beefy body.
And the Hereford calves, always from a Hereford bull and Hereford cow, show the same red bodies and white faces. But why breed Herefords?
Because they're the best beef-producing breed of cattle. Quick maturing and good converters of all types of feed.
But for milking, Frank Smith keeps another breed of cattle.
A Friesian herd, dairy herd, which are supposed to be the best milk producers. The Friesian is the main characteristics are 4 white feet, a black and white body, a white tip to the tail, and usually a black face with a white diamond on the forehead. Friesians are the best milk producers.
But Frank doesn't want too many Friesian calves, so he mates his Friesian cows to a Hereford bull. The beef characteristic of the Hereford is passed on, so Hereford Friesian calves are good for beef. They're also easy to spot.
The white face is a dominant characteristic of a Hereford crossed with a Friesian.
So the white-face gene from the Hereford bull must be dominant, and the black-face gene from the Friesian cow recessive. The cross calf on the left is easily distinguished from the pure Friesian on the right. So when a cross calf is taken to market, what do the buyers think of it?
They usually pay more money for it, and because they know it is by a Hereford bull and will be a good beef producer.
Knowing which genes are dominant and which are recessive can help a farmer breed and buy the best calves. In humans, genetics is most important for families with inherited diseases. At University College Hospital in London, they are studying the inherited disease called thalassemia.
Well, thalassemia is a severe inherited blood disease of children. And it affects them sometime in the middle of the first year of life. They're perfectly normal at birth and usually up to about 6 to 9 months of age. But then they begin to get severely anemic. Now, the problem really is that they can't make their own blood. In summary, the treatment of the disease really is that the children have to live on borrowed blood for the rest of their lives.
But how can children get the disease when their parents seem healthy?
Well, it's inherited in a recessive way. That means that it's inherited from couples of people who carry it but don't know they carry it because they've not got any symptoms. We call them symptomless carriers.
Carriers have one gene for normal blood and one for thalassaemic blood, but the normal blood gene is dominant, so they're healthy. If the carrier parents both pass on their normal blood genes, their child will be healthy and not even a carrier. If one passes on a normal blood gene and the other a thalassaemic blood gene, the children will be carriers, but they'll be healthy, as the normal gene is dominant. But if both parents pass on their thalassaemic blood genes, their child will have the disease. So this happens in 1/4 of the cases. Can carriers be detected?
Well, we're very fortunate with thalassemia because, by contrast with most inherited diseases, it is possible to find out if you're a carrier. So that, from the person's point of view, all they need to do is know that there's a possibility that they could be a carrier. And then when the right time comes, like when they're thinking of getting married, they can go and ask for a blood test.
Then what happens if both the man and the woman are carriers and the woman becomes pregnant?
Well, it is possible now to have tests during the pregnancy to see if the baby is normal, which of course happens 3 out of 4 times, or if it's affected by the disease, which happens in 1/4 of these pregnancies.
The medical team have developed a new technique for testing women who are only 8 weeks pregnant.
It's a little bit ungraceful for the lady because we've got to approach through the vagina and the cervix, so she has to lie on her back. And then we have a very good ultrasound machine which produces a very clear picture of the early pregnancy.
Using the ultrasound picture, the surgeon can see inside the womb to remove some of the tissue surrounding the unborn baby. The tissue cells contain the baby's chromosomes. The blood-making genes on the chromosomes can be examined to see if they're normal or thalassaemic. Why is Dr. Modell so keen that parents should know there are tests available?
Because the whole value of preventing genetic disease by this approach depends on knowledge. And it's not the knowledge of the doctors, it's the knowledge of the people about that genetic disease exists, that it could be them, that everybody can be a carrier of one or another inherited condition, however healthy they may feel. They've got to know that in order to ask to be tested.
Bassos and Maria Servidis only discovered they were both carriers of thalassemia when Maria was pregnant with their first child.
Well, after we found out that we were both carriers, we discussed what to do in the event that the baby would be diseased. And we decided that we would abort the baby. It didn't take us very long, actually, to reach that conclusion.
But luckily the baby was normal.
Oh, thank goodness it was all right. And Stephanie is not even a carrier. She's absolutely clear of the disease, which is only 25% each case to have a clear baby. And she was one of the lucky ones.
And when should people find out if they're carriers?
Well, it's very, very important to find out at least after they get married, but before they're pregnant. So they get used to the idea a little bit, not come to the late stage of the pregnancy and face the problem the last minute.
It's good to be able to make your own decisions, rather than to suddenly find you have a baby that is ill or diseased and have to suffer for the rest of your life, and for that child to have to suffer for the rest of its life.
But scientists knew that Stephanie was perfectly normal by looking at her genes. But it's one thing to know about genes, quite another to change them. And that's what's happening in San Francisco at the University of California Medical School. People from all over the world are working on altering genes, a process called genetic engineering.
Well, genetic engineering is really the ability to take pieces of genetic material from different organisms and put them together in ways or combinations that don't appear to have occurred naturally.
For Gerard, work starts by finding some genetic material, the stuff which makes up the chromosomes. The key substance is an enormously complicated chemical called deoxyribose nucleic acid, known as DNA. A few years ago, scientists in these laboratories discovered that certain enzymes could cut the DNA molecule into bits. Then, using other enzymes, DNA bits from different living things could be joined together. Genetic engineering had been achieved. It's still early days, but the technique could be extremely useful. Using human cells, an enzyme can cut out a piece of the human DNA. In bacterial cells, where some of the DNA exists in rings, the same enzyme can chop out a piece of the bacterial DNA. The piece of human DNA can then be stuck into the bacterial DNA ring. Now the bacterial cell will follow the instructions of the human DNA as well as its own and make a product that a human could make. As bacteria multiply very quickly, vast quantities of important human products can be made.
For an example, the disease haemophilia, in which people, when they're cut, their blood refuses to clot and they will just bleed to death. This is because they lack a certain protein called factor VIII. Now, factor VIII is made in very, very small quantities in normal human beings and not at all in haemophiliacs, which is why they bleed to death. We've been able to take the factor VIII gene and to put it into bacteria, and the bacteria act as a sort of factoring, and they make vast quantities of factor VIII. We can then purify the factor VIII from a bacterial culture and give it to people who don't make it normally, and thereby cure their disease.
Working with so many bacteria, is genetic engineering dangerous?
No, I don't think so. When the science first began, or the technology first began, there was a lot of scare and a lot of worry, mainly amongst the scientific community, about the possibilities of generating new forms of bacteria with new genetic makeups that could cause terrible diseases. By and large, throughout all the amount of genetic engineering, none of that's occurred. It's all done in rooms where everything is kept sterile and clean, and every bacterial culture that's made is autoclaved. That means it's heated in an oven until it's dead. Certainly we're not going to develop any Frankenstein monsters. In the future, obviously one is more excited by the prospect of taking embryos that one knows are going to suffer from a certain genetic disease and taking out the defective gene from them and putting back in a normal gene via recombinant DNA genetic engineering techniques, and thereby correct the illness at the very, very early stage of differentiation of the embryo, and therefore give rise to normal human beings.
Is it exciting work?
I think there must be few areas of science that are more exciting. In fact, I don't believe there are any. The areas of high-energy physics, I suppose, are as exciting, but in terms of the pace at which things move, in terms of the possible applications of one's work at the moment in cancer biology, which is, for instance, going on at this laboratory at the moment, in terms of medical science, in terms of corrective and preventative medicine, I really don't believe if you're interested in disease and human beings and trying to cure these things, I don't believe there's any science or any particular aspect of science that is more interesting, no.